Life exists in many forms, from towering giant redwood trees to the smallest microbes that measure just a few billionths of a meter. Throughout history, humans have wondered about the origins of life and what defines it. Our understanding of life has evolved significantly since the invention of the microscope. In the 17th century, scientists observed microscopic life and developed the cell theory, which posits that cell is the basic unit of life. All living organisms are made up of at least one cell, and that cells arise from existing cells. Cells, while sharing certain characteristics, can differ greatly from one another. The two primary types of cells are prokaryotic cells, which do not have a nucleus, and eukaryotic cells, which possesses a well-organized membrane-bound nucleus. Each cell type showcases a remarkable variety in structure, function, and metabolic processes. This chapter will delve into the historical discoveries that have shaped our current understanding of microbes, their origins, and their significance in human disease. Additionally, we will examine the unique structure present in prokaryotic cell. In the program, also have a separate Biology 8420, Understanding Cell Biology, based on eukaryotic cell. We will not discuss the section 3.4, Unique Characteristic of Eukaryotic Cells, in this course. Microorganisms exhibit a diverse range of sizes and shapes that can be seen under a microscope. These variations are not just superficial, they also extend to their metabolic functions, which are often not visible to the naked eye. This means that while we can observe the physical characteristic of microorganisms, understanding their biological processes require more in-depth analysis. For instance, some microorganisms are spherical, while others may be rod-shaped or spiral. This diversity in morphology can be linked to their ecological roles in the environment in which they thrive. Additionally, metabolic capabilities can differ significantly among various types of microorganisms. Some are capable of photosynthesis, converting the light energy into chemical energy, while others may rely on organic compounds for energy, showcasing the different ways these organisms adapt to their surroundings. Moreover, these metabolic differences play a crucial role in nutrient cycling and energy flow within the ecosystem. By studying these microorganisms, scientists can gain insights into their function and interaction within their habitats. Understanding both the visible and invisible characteristic of microorganisms is essential for appreciating their complexity and importance in the biosphere. For thousands of years, humans have pondered, what is the origin of new life? This question has been explored by religion, philosophy, and science alike. Among the earliest theories was spontaneous generation, which originated from the ancient Greeks and remained a widely accepted belief into the Middle Ages. This section will explore the theory of spontaneous generation, examining the support and opposing arguments surrounding it. The discussion will highlight key points that illustrate the historical context and scientific debates related to the theory, providing a comprehensive overview of its significance in understanding biological origins. The Greek philosopher Aristotle was one of the first to propose the theory of spontaneous generation, which suggested that life could emerge from non-living matter. He believed that life originated from materials containing pneuma or vital heat. Aristotle cited examples such as a sudden appearance of fish in the puddles of water, arguing that this phenomenon supported this theory of life arising from lifeless environment. By the 17th century, the theory of spontaneous generation continued to be debated as scientists sought to prove evidence for or against it. Observations were made, such as frogs seemingly appearing along the banks of Nile during the annual floods, mice appearing in stored grain when conditions allowed. Jean-Baptiste van Helmont, a Flemish scientist of that time, speculated that mice could emerge from rag and wheat kernels left in an open container for 3 weeks, not recognizing that these conditions actually provided food and shelter for mice to thrive. In contrast, Italian physician Francesco Redi conducted a pivotal experiment in 1668 that challenged the idea of spontaneous generation. He tested whether maggots could arise from exposed meat by using 6 containers, 2 open to the air, 2 covered with gauze, and 2 tightly sealed. Reddy's results showed that maggots developed only in the open jars, while the gauze-covered and sealed jars remained free of maggots. This experiment provided evidence that maggots arise from flies, supporting the idea that life does not spontaneously generate from non-living matter. In 1745, John Needham conducted an experiment where he boiled broth mixed with plant or animal matter to eliminate existing microbes, then sealed the flask. After a few days, he found the broth cloudy and filled with microscopic organisms, concluding that the microbes arose spontaneously. However, it is likely that Needham did not boil the broth long enough to kill all pre-existing microbes present. Disagreeing with Needham, Lazzaro Spallanzani performed numerous meticulous experiments using heated broth. His experiment involved both sealed and unsealed jars containing infused broth. Spallanzani observed that the heated sealed flask remained clear and free from spontaneous growth unless exposed to air, indicating that microbes were introduced from the environment. In defense of his findings, Needham claimed that life force was destroyed during Spallanzani's prolonged boiling, preventing new life force from entering a sealed flask and thus causing spontaneous generation. The debate over spontaneous generation persisted into the 19th century, with scientists divided on the topic. To resolve the issue, the Paris Academy of Sciences offered a prize for a definitive answer. Louis Pasteur, a distinguished French chemist known for his work on microbial fermentation and wine spoilage, took the challenge. In 1858, he filtered air through a gun cotton filter and discovered an abundance of microorganisms on the cotton, indicating that exposure of broth to air did not introduce a life force, but rather airborne microorganisms. In his subsequent experiment, Pasteur devised swan neck flasks, which had long twisted necks. He boiled broth in this flask to sterilize it, allowing air exchange while preventing airborne microorganisms from entering. Pasteur predicted that as long as the flask necks remained intact, the sterilized broth would stay uncontaminated. He demonstrated that if the necks were broken, microorganisms could enter, leading to microbial growth. This design effectively showcased that life did not spontaneously generate, but came from existing microorganisms. Pasteur's experiment conclusively disproved the theory of spontaneous generation, earning him the Alhumbert Prize from the Paris Academy of Sciences in 1862. In a notable lecture in 1864, he expressed the principle omni vivum ex vivo, meaning life only comes from life, and emphasized the significance of his own swan neck flask experiment. He asserted that the doctrine of spontaneous generation would never recover from the decisive evidence provided by his work, a prediction that has held true to this day. The term cells was first introduced by the English scientist Robert Hooke in 1665, when he examined cork through a compound microscope. He observed small chambers that resemble honeycomb or small boxes or bladders of air. Hooke noted that each cavern bubble or cell was distinct from one another. However, he was unaware that these cork cells were long dead and did not contain the internal structure characteristic of a living cell. Although Hooke's observations were groundbreaking, the true significance of cells as the fundamental unit of life was not recognized until much later. In 1838, German botanist Matthias Schleiden conducted extensive microscopic examination of plant tissues and concluded that plants were composed of cells. He found plant cells easy to visualize due to their thick cell walls, but mistakenly believed that cells formed through crystallization rather than division. In 1839, Theodor Schwann, a German physiologist, conducted similar microscopic studies on animal tissues. After discussing his findings with Schleiden, Schwann identified the similarities between the plant and animal cells. This collaboration contributed to the development of the cell theory. which asserts that all living organisms are composed of cells, marking a significant advancement in understanding of the biology. In 1850s, two Polish scientists residing in Germany advanced the idea of cell theory, leading to what we now know as modern cell theory. In 1852, Robert Remak, a notable neurologist and embryologist, provided compelling evidence that cells originate from other cells through cell division. This concept, however, faced skepticism from many scientists. 3 years later, Rudolf Virchow, a highly regarded pathologist, wrote an editorial essay titled Cellular Pathology, which helped to popularize the theory with the Latin phrase omnis cellula a cellula. All cells arise from cells, essentially representing the second principle of modern cell theory. Due to the similarities between the works of Virchow and Remak, there is ongoing debate regarding which scientists should be credited with establishing cell theory. For further insights into this controversy, refer to the following Eyes on Ethics feature. As scientists advanced in understanding the roles of cells within the plant and animal tissues, they also began to investigate the internal structure of cells. In 1831, Scottish botanist Robert Brown made significant contributions by being the first to identify nuclei in plant cells. This discovery laid the groundwork for further exploration. By the early 1880s, German botanist Andreas Schimper expanded on this knowledge by describing chloroplasts and their essential function in photosynthesis, particularly in starch formation. He noted that chloroplasts could divide independently of the cell nucleus, highlighting a unique characteristic of these organelles. In 1905, Russian botanist Konstantin Mereshkovsky proposed that the ability of chloroplasts to reproduce independently suggested that they may have originated from ancestral photosynthetic bacteria that lived symbiotically within the eukaryotic cells. His idea extended to the nucleus of plant cells as well, marking the introduction of the endosymbiotic hypothesis. This hypothesis provided a potential explanation for how complex eukaryotic cells evolved from simple ancestral bacteria, setting the stage for further scientific inquiry into cellular origins. The endosymbiotic hypothesis gained traction through the work of American anatomist Ivan Wallin, who conducted experiments comparing mitochondria, chloroplast, and bacteria. In 1920s, Wallin published several papers, including collaboration with Mereshkovsky, which supported the hypothesis. He claimed to culture mitochondria outside of their eukaryotic host, although many scientists discredited his findings, attributing them to bacterial contamination. Modern genome sequencing has since reinforced the skepticism, revealing that many mitochondrial genes have been transferred to the host cell's nucleus, which restricts mitochondria from surviving independently. Wallin's endosymbiotic hypothesis was largely overlooked for 50 years due to the lack of awareness that organelles contain their own DNA. However, the discovery of mitochondrial and chloroplast DNA in the 1960s revived the interest in this hypothesis. Lynn Margulis, an American geneticist, published her ideas on the origin of these organelles in 1967, building upon advances in microscopy that allowed for the differentiation of prokaryotic and eukaryotic cells. In her work, Margulis reviewed existing literature and presented evidence from microscopy, genetics, molecular biology, fossils, and geology to argue that eukaryotic organelles like mitochondria and chloroplast originated from prokaryotic cells. The endosymbiotic theory, which posits that mitochondria and chloroplast originated from prokaryotic cells, forming symbiotic relationship within the eukaryotic host, gained acceptance due to increasing genetic evidences from DNA sequencing. Initially proposed by Lynn Margulis and expanded upon her 1981 book Symbiosis in Cell Evolution, this theory highlights the endosymbiosis as a key factor in the evolution of organisms. Genetic sequencing has revealed that mitochondrial and chloroplast DNA closely resembles that of bacteria, both in sequence and structure, although these organelles have reduced DNA due to gene transfer to the host nucleus. Additionally, their ribosomes are similar to bacterial ribosome and they replicate through binary fission akin to bacteria, rather than through eukaryotic mitosis. Modern observations have also identified bacterial endosymbionts in various eukaryotic cells, such as those found in the guts of insects like cockroaches and certain protists. Before the 17th century and the discovery of microbes, various theories existed regarding the origins of diseases. Among these, the ancient Greeks proposed the miasma theory, which suggested that diseases arose from harmful particles released by decomposing matter such as sewage. These particles were believed to infect people nearby, and this theory was used to explain outbreaks like the Black Death that devastated Europe during the Middle Ages. In 1546, Italian physician Girolamo Francastoro advanced the idea that diseases could be spread by seed-like spores through direct contact, contaminated clothing, or the air. His essay, De Contagione et Contagiosis Morbis, laid a groundwork for what we now know as germ theory, which posits that microorganisms are the cause of many diseases. However, during the 16th century, Francastoro's ideas did not gain traction and were largely overlooked until they were revived in the 19th century. In 1847, Hungarian obstetrician Ignaz Semmelweis made a significant observation related to maternal health. He noted that mothers who gave birth in wards with the physicians and medical students had much higher rates of puerperal fever compared to those attended by midwives. Semmelweis linked this discrepancy to the practice of medical students conducting autopsies and then examining living patients without washing their hands in between, suspecting that they were transmitting disease. Although Semmelweis did not identify the exact cause of puerperal fever, he proposed that hand washing with chlorinated lime water could significantly reduce infection rates. When this practice was adopted, the maternal mortality rate fell to the same low level observed among midwives. Despite the success of his finding, many in the medical community dismissed Semmelweis's work, as it challenged the established norm of his time, leading to a slow adoption of hand hygiene practices in healthcare and settings. John Snow was an influential English physician recognized for his contribution to anesthesia and medical hygiene, and he is regarded as a pioneer of modern epidemiology and early germ theory. His significant work in tracing the source of a cholera outbreak in London's Soho to a specific public water pump led to the essential reforms in the city's water and waste systems. His work, which is recognized as the first known epidemiological study, demonstrated that cholera bacteria could be transmitted through drinking water, leading to the first public health response to an epidemic. The John Snow Public House in Soho, London, was originally known as the Newcastle upon Tyne, dates back to the 1870s, and was reconstructed at the location of the water pump identified by John Snow as the source of a cholera outbreak in 1854. In 1954, a century after the pump's handle's removal, the pub was renamed as the John Snow Public House. The pub sign was unveiled in May 1955 by Sir Austin Bradford Hill, the president of the Epidemiology and Public Health section of the Royal Society of Medicine. Together with Ignaz Semmelweis's promotion and hand washing, Snow's findings challenged the prevailing miasma theory of disease transmission, highlighting that disease could spread not only through the air, but also through contaminated materials. In 1856, Louis Pasteur made a significant discovery while studying the spoilage of beer and wine, unveiling the role of microorganisms in fermentation. Through his Swan Lake flask experiment, he demonstrated that airborne microbes rather than spontaneous generation were responsible for food spoilage. These pivotal findings led him to propose that if the microbes could spoil food and facilitate fermentation, they might also have responsible for infection. This idea laid the groundwork for the germ theory of disease, fundamentally challenging the understanding of how disease spread. At the same time, British surgeon Joseph Lister sought to understand the causes of post-surgical infection, which were resulting in a high mortality rate among the surgical patients. Despite the evidence, many physicians were skeptical about the impact of microbes from their hands or clothing or surgical wounds. Drawing inspiration from the findings of Semmelweis and Pasteur, Lister advocated for strict hand washing and cleanliness during surgeries. In 1867, he introduced the use of carbolic acid, which is phenol, as a disinfectant during operations, significantly reducing the rates of establishing a new standard in surgical practice. The well-known mouthwash Listerine is used worldwide. The name Listerine has a fascinating origin that highlights its historical importance, as it is named after the last name of Joseph Lister. Shortly after, Robert Koch developed a set of criteria known as Koch's postulates, which linked specific diseases to specific microbes. His work enabled the identification of pathogens responsible for diseases such as anthrax, tuberculosis, and cholera. Reinforcing the connection between the germs and illness, Koch's assertion of one microbe, one disease represented a major shift away from the outdated miasma theory towards the germ theory of disease, marking a transformative period in medical sciences. The slide presents a clear and detailed timeline highlighting significant contributions to the germ theory of disease. It outlines key discoveries and advancements that have shaped our understanding of how germs causes illness, providing a comprehensive overview of evolution of this vital scientific theory. Cell theory establishes that the cell is the fundamental unit of life. Yet, cells exhibit considerable diversity in size, shape, structure, and function. At their core, all cells share a few essential components. Cytoplasm, which is a gel-like substance composed of water and necessary dissolved chemicals. A plasma membrane that encloses the cytoplasm. Chromosomes that carry the genetic information, and ribosomes, which are responsible for protein synthesis. These components are crucial for the cell's growth and function. Cells can be broadly categorized into two main types, prokaryotic and eukaryotic cell, characterized by distinct structural differences. Prokaryotic cells do not have a nucleus or a surrounding nuclear membrane, as we have already learned, and they typically contain a single circular chromosome located in the region called nucleoid. In contrast, eukaryotic cells possess a nucleus enclosed by a complex nuclear membrane, housing multiple rod-shaped chromosomes, and fundamental distinction underlies the complexity and organization seen in the eukaryotic cells compared to prokaryotic cells. Eukaryotic cells encompasses both plant and animal cells, while prokaryotic microorganisms are found within the domain of archaea and bacteria. This classification highlights the variety of cellular structures present in different types of organisms. For instance, certain microorganisms may consist of prokaryotic cells, while others may have eukaryotic cells, illustrating the diversity of life forms and their cellular compositions. Inside a cell, various structures operate similar to organs in human body, which tailored to fulfill specific functions. While some structures in prokaryotic cells are comparable to those in eukaryotic cell, others are unique to prokaryotes. Generally, eukaryotic cells are larger than prokaryotic cells, necessitating compartmentalization of diverse chemical processes within specialized membrane-bound organelles. Prokaryotic cells, on the other hand, usually lack these organelles, but may possess inclusions that help organize their cytoplasm. The next section will provide more detailed description of these structures commonly associated with the prokaryotic cell. Prokaryotic organisms generally exhibit similar shell shapes, known as cell morphologies. Despite the identification of thousands of prokaryotic species, only a few morphologies are frequently observed under a microscope. These common morphologies include specific shapes that can be illustrated and named on a slide. Understanding these typical cell forms is essential for studying prokaryotic biology and helps in identifying various prokaryotic organisms through their visual characteristic. The slide figures serves as a visual reference for these commonly seen prokaryotic cell morphologies. Prokaryotic cells of the same species can exhibit various arrangements based on their cellular shape and the orientation of cell division. These distinctive groupings can arise from the cells divided in different planes. The arrangement of these cells is important for understanding their structure and function. For instance, some cells may cluster in chains, while others might form clusters or pairs. Depending on how they divide, these common arrangements help in identifying, classifying prokaryotic organisms. The slide figure shows the example of these typical cellular formations. A single coccus is a single cell, whereas Diplococcus, it divides in one plane, so they are two. Tetrad, they are developing in two planes, and that's why there are four. Streptococcus, they are dividing in one plane in chains. Staphylococcus can divide in multiple planes, which is forming a cluster of cocci. Bacillus is a single rod-shaped bacteria, and Diplobacillus they are two together. Streptobacillus is a chain of these bacillus, whereas the palisades are V or L-shaped structures. Let's take a look at the osmotic environment and the cell behavior before getting into the detail of the prokaryotic cell structure. Cells exist in various osmotic environments that significantly influence their behavior and function. The three primary types of osmotic environment are isotonic, hypotonic, and hypertonic, each affecting the movement of water across the cell membrane through osmosis. Prokaryotic cell morphology is primarily maintained by the cell wall and cytoskeletal elements, with the cell wall serving as a protective structure that envelopes the cell membrane to guard against the osmotic pressure changes. Osmotic pressure arises from the solute concentration differences across a semi-permeable membrane, where water can pass through while solutes cannot. Water diffuses from an area of lower solute concentration to one of the higher concentration until the equilibrium is reached, a process known as osmosis. This can lead to significant osmotic pressure effects on cells when their external environment fluctuates. In an isotonic solution, the concentration of solutes outside the cell is equal to that inside the cell. This balance means that the water moves in and out of the cell at equal rates, resulting no net change in the cell volume. Cells in isotonic environment tend to maintain their normal shape and function optimally. This environment is crucial for maintaining homeostasis, particularly in human blood, where isotonic conditions are vital for healthy and cellular activity. A hypotonic solution has a lower concentration of solutes compared to the inside of the cell. When a cell is placed in a hypotonic environment, water flows into the cell in an attempt to balance the solute concentrations. This influx can cause the cell to swell and potentially burst, a process known as lysis or osmotic lysis. Plant cells, however, benefit from hypotonic environment as they maintain turgor pressure, which keeps them rigid and structurally sound. Conversely, a hypertonic solution has a higher concentration of solutes than the cell's interior. When cells are in a hypertonic environment, water exits the cell to the surrounding solution, leading to the cell shrinkage or crenation. This condition can impair cellular function and is particularly detrimental to animal cells. In plants, hypertonic environment can lead to plasmolysis, where the cell membrane pulls away from the cell wall, comprising the plant's structural integrity. Tonicity refers to how well a cell can handle changes in the osmotic pressure. Cells with a cell wall are more capable of managing slight shifts in osmotic pressure, helping them to keep their shape. In hypertonic conditions, cells without a cell wall may lose water, leading to crenation or shriveling. Their plasma membrane pulls in and takes on a scalloped appearance. In contrast, cells with cell wall experience plasmolysis instead of crenation. During plasmolysis, the plasma membrane shrinks away from the cell wall, causing reduction in internal volume, but the cell wall stays intact, allowing the cell to retain some shape and stability for a while. Similarly, cells that do not have a cell wall are more likely to burst in the hypotonic environment. The existence of a cell wall enables the cells to maintain its shape and structure longer before it bursts. The first bacterial internal component is cytoskeleton. Initially, it was believed that bacteria did not possess a cytoskeleton, which is a crucial part of the eukaryotic cells. However, in the past 2 decades, researchers have identified bacterial filaments composed of proteins that are similar to the cytoskeletal proteins present in eukaryotes. Furthermore, studies have shown that bacterial cytoskeleton is vital for maintaining cell shape, facilitating cell division, and ensuring the integrity of the cell wall. FITZ or FTSZ is homologous to eukaryotic protein tubulin, forms a ring-like structure in the middle of the cell during the cell division, attracting other proteins to the area to construct a septum that will eventually separate and resulting in 2 daughter cells. MREB, homologous to the eukaryotic protein actin, is found in bacillus and spiral-shaped bacteria and plays an essential role in cell shape formation. MREB assumes a helical configuration running the length of the cell and dictates the activities of peptidoglycan synthesis machinery, assuring a non-spherical shape. Crescentin, a homologous to the eukaryotic proteins laminin and keratin, is found in spiral-shaped bacteria with a single curve. The protein assembled lengthwise in the inner curvature of the cell, bending the cell into its final shape. The nucleoid is a defining characteristic of prokaryotic cell, distinguishing them from eukaryotic counterparts. Unlike eukaryotes, which possesses a membrane-bound nucleus, prokaryotes contain their genetic material within an irregularly shaped region called nucleoid. This region is devoid of a surrounding membrane, allowing for direct interaction between the genetic material and the cytoplasm, which is crucial for the various cellular processes. The primary component of the nucleoid is the chromosomal DNA, typically consisting of a single circular double-stranded DNA molecule. This chromosomal DNA contains the essential genetic information required for the organism's growth, reproduction, and metabolic function. In addition to chromosomal DNA, prokaryotic cells often harbor plasmids, small extrachromosomal DNA molecule that can carry genes conferring advantageous traits such as antibiotic resistance. Plasmids play a significant role in horizontal gene transfer, contributing to the genetic diversity and adaptability in prokaryotic populations. The organization of DNA within the nucleoid is facilitated by a range of nucleate-associated proteins or NAPs, which aid in the compaction and structural maintenance of the DNA. These proteins promote supercoiling, a process that involves the twisting of the DNA double helix, thereby allowing for efficient packaging within the limit of the cellular space. Supercoiling is critical for processes such as transcription and replication, as it influences the accessibility of the DNA to the necessary molecular machineries. During cell division, the nucleoid undergoes a precise replication process, ensuring that each daughter cell inherits a complete functional copy of the genetic material. The simplicity of the nucleoid's structure enables rapid replication and cell division, which are hallmarks of prokaryotic life, particularly in the environments where growth rates are elevated. Prokaryotic cells can also possess the extrachromosomal DNA, as we have said, which is DNA not included in the main chromosome. This extrachromosomal DNA can exist in structures called plasmids, which are small circular double-stranded DNA molecules. Cells containing plasmid can have hundreds of these within a single cell. While plasmids are primarily found in bacteria, they have also been identified in archaea and eukaryotic organisms. Plasmids frequently carry genes that provide beneficial traits such as antibiotic resistance, making them vital for the organism's survival. We will explore plasmids in greater detail in the section on mechanism of microbial genetics. All living cells make proteins, and organisms in the three domains of life have ribosomes, which are the structures that help in protein production. However, ribosomes vary in structure across these domains. Ribosomes are made up of proteins and ribosomal RNA, rRNA. Prokaryotic ribosomes are known as 70S ribosome due to their size, while eukaryotic cytoplasmic ribosomes are larger, classified as 80S. The S refers to the Svedberg unit, sedimentation coefficient, a measurement used to determine how fast particles settle in an ultracentrifuge depending on their size, shape, and surface characteristics. Even though bacterial and archaeal ribosomes are both 70S, they contain different proteins and rRNA, with archaeal ribosomes being more similar to eukaryotic ribosomes than to those in bacteria. Please note that Svedberg units are not additive. 30S small subunit and the 50S large subunit of prokaryotic ribosome make a 70S complete ribosome and not 80S. Similarly, the 80S eukaryotic ribosome is made up of a smaller 40S and larger 60S subunit. Prokaryotic cells, which are single-cell organisms, often live in unpredictable environments. They can store extra nutrients in special structure within their cytoplasm called inclusions. This storage is helpful because it prevents high osmotic pressure, which happens when too many solutes build up in the cell. Different types of inclusions can hold substances like glycogen and starches, which provide carbon for energy. One type, called volutin granules or metachromatic granules, due to their unique staining properties, store polymerized inorganic phosphate. This phosphate plays a role in metabolism and helps create biofilms. Some microbes that have volutin granules include an archaeon Methanosarcina, the bacterium Corynebacterium diphtheri, and unicellular eukaryotic alga Chlamydomonas. Another type of inclusion, sulfur granules, is found in sulfur bacteria from the genus Thiobacillus. These granules contain elemental sulfur, which the bacteria use for their metabolic process. Sometimes specific kinds of inclusions are encased by a phospholipid monolayer containing proteins. An example of such an inclusion is polyhydroxybutyrate or PHB, which can be produced by Bacillus and Pseudomonas species. In industrial application, PHB is utilized as a source of biodegradable polymers or bioplastics. The slide figure represents various types of these inclusions. Some prokaryotic cells have a various type of inclusion that serve roles beyond just storing nutrients. For instance, certain prokaryotic cells create gas vacuoles, which are clusters of small protein-like vesicles filled with gas. These gas vacuoles enables the prokaryotic cells that produce them to change their buoyancy, allowing them to move within a water column. Magnetotactic bacteria, like Magnetospirillum magnetotacticum, contain magnetosomes, which are inclusions made up of magnetic iron oxide or iron sulfide encased in a lipid layer. This feature helps the cells align with a magnetic field, facilitating their movement. Cyanobacteria such as Anabaena cylindrica and bacteria Halothiobacillus neapolitanus generate carboxysome inclusions. Carboxysomes consist of outer shells made up of thousands of protein subunits, with their interior filled with ribulose 1,5-bisphosphate carboxylase oxygenase or Rubisco and carbonic anhydrase, both of which are essential for carbon metabolism. Furthermore, some prokaryotic cells have carboxysomes that group together related to enzymes in one area. These structures are regarded as proto-organelles because they compartmentalize vital compounds or chemical reactions similar to many eukaryotic organelles. Endospores represent a remarkable adaptation in bacterial cells. Although categorized under bacterial internal components, it's essential to understand that an endospore is not merely an internal or external structure. Rather, it signifies a transformation of the cell into different state. Bacterial cells typically begin as vegetative cells, performing essential life functions such as metabolizing and reproducing. However, when faced with harsh conditions like desiccation, extreme heat, or competition, these cells may convert into endospores if they possess the capability to achieve remarkable resistance against various substances and environmental conditions. Bacterial endospore consists of multiple layers. At the center lies the core, which houses the nucleoid, ribosome, and cytoplasm in a very dehydrated stage. The core typically consists of 25% of water present in a vegetative cell, enhancing its heat resistance. Small soluble protein, known as SASPs, further protect the DNA by stabilizing and preventing degradation. The stabilization of DNA is additionally supported by dipicolonic acid, which forms a complex with calcium or calcium DPA that intercalates between the DNA bases. Surrounding the core is an inner membrane that acts as a barrier to chemicals, followed by a cortex, a robust layer of peptidoglycan with reduced cross-linking compared to that of a vegetative cell. Encasing the cortex is an outer membrane, and finally, several proteinaceous spore coats provide an extra layer of protection against environmental stresses, including chemicals and enzymes. Here you can see an electron micrograph, and here you can see that a Bacillus species, which is undergoing sporulation, and here the spores are visualized by malachite green spore stain, as we have discussed in the other section. The endospore develops within the vegetative cell, leading to some debate about whether it is an internal structure. Please remember, a vegetative cells in microbiology or biological sciences mean a cell which is metabolically active. Vegetative state of human beings often considered as when people go into the coma. That is not the case in here, so just make sure that here we understand a vegetative cell is a fully functional, metabolically active cell. Eventually, the vegetative cells breaks down, releasing the endospore into the environment, which raises the question of whether it then becomes an external structure. Only a select few gram-positive bacterial genera can form endospores, which equip the cells with a resistance against numerous adverse conditions, including starvation, temperature extremes, desiccation, UV radiation, and exposure to chemicals and enzymes. While the vegetative cells is active, engaging in growth and metabolism, as we just mentioned, the endospore serves as a dormant phase of the cell. This dormant state enables survival under challenging conditions, but it does not allow for growth or reproduction. Sporulation is the process through which a vegetative cell converts into endospores. This typically occurs when nutrients are low or when environmental conditions are not suitable. It starts with development of a septum within the vegetative bacterial cell. This septum divides the cell unevenly, creating a DNA forespore that is distinct from the mother cell. The forespore, which will become the core of the endospore, is a copy of the cell's chromosome and is encased by a second membrane. As the process continues, the cortex forms around the forespore by layering calcium and dipicolonic acid between the membranes. A protective protein spore coat is then created around the cortex while the DNA of the mother cells breaks down. The endospore matures with the addition of the outermost layer called the exosporium. Finally, the endospore is released when the mother cell disintegrates, marking the completion of sporulation. In this section, we are going to talk about the bacterial membranes, and the cell walls. As we cover membrane and membrane transportation extensively in Biology 8420, Understanding Cell Biology, we will only discuss the unique characteristic of the plasma membrane in bacteria and archaea. The structures that surround the cytoplasm and internal parts of a cell are called cell envelope. In prokaryotic cells, the components of the cell envelope differ based on the types of cell and organism. Most prokaryotic cells have a cell wall, though its composition can vary. All cells, whether prokaryotic or eukaryotic, possess a plasma membrane, also known as the cytoplasmic membrane or cell membrane, that has selective permeability, which permits certain molecules to enter or exit the cell while preventing others from passing through. The plasma membrane's structure is commonly explained using the fluid mosaic model. This model highlights how the components of the membrane can move freely within its plane and emphasizes the diverse arrangement of these components, which include various lipids and proteins. Most bacterial and eukaryotic cells have a plasma membrane that is a bilayer, primarily made of phospholipids linked by ester bonds and protein. Both phospholipids and proteins within the membrane can move laterally across the surface and between the two phospholipid layers. There are several characteristics of the plasma membrane that are unique to archaea, setting them apart from other domains. One such characteristic is the chirality of the glycerol linkage between the phospholipid head and the side chain. In archaea, it is in L-isomeric form, while bacteria and eukaryotes have the D-isomeric form. A second difference is the presence of an ether linkage between the glycerol and its side chain, as opposed to the ester-linked lipids that is found in bacteria and eukaryotes. The ether linkage provides more chemical stability to the membrane. A third and fourth difference are associated with the side chains themselves. Unbranched fatty acids in bacteria and eukaryotes, while isoprenoid chains are found in archaea. These isoprenoid chains can have branching side chains as well. Some prokaryotic cells, including cyanobacteria and photosynthetic bacteria, possess membrane structure that allow them to carry out photosynthesis. These structures are formed by infolding of the plasma membrane, which houses photosynthetic pigments like green chlorophylls and bacteriochlorophyll. In cyanobacteria, these membrane structures are referred to as thylakoids. In photosynthetic bacteria, they are known as chromatophores, lamellae, and chlorosomes. Cyanobacteria, often referred to as blue-green algae, have extensive thylakoid systems that contain chlorophyll and other pigments. These pigments capture light energy, which is then converted into chemical energy through a series of electron transport chains. Purple bacteria, on the other hand, utilize bacteriochlorophyll and have different types of photosynthetic membranes. These bacteria can have either folded membranes or vesicular structure that facilitate light capture and energy conversion. The arrangement of these membranes is crucial for maximizing the efficiency in varying light conditions. If a cell wall is present in an organism, not just in bacteria, it is found outside of the cell membrane. It's an additional layer that typically provides some strength that the cell membrane lacks by having semi-rigid structure. Bacterial morphology is primarily influenced by the structure of the cell wall. Tenoricutes was a phylum of gram-negative bacteria consisting of cells bounded by a plasma membrane. Its organisms differ from the other bacteria in that they lack cell walls. This phylum was formerly the class Mollicutes. Mollicute is now the sole class in the phylum Tenoricutes. This classification was introduced in 2011. This phylum is the only phylum among the bacteria without a cell wall. In 2021, the Tenoricutes phylum was again renamed to Mycoplasmatota, which is the most current classification. In general, they are called mycoplasma. Depending on their nutrient and host requirements, they are classified as a saprophyte or plant, fungal, insect, and animal mycoplasma. They are highly pleomorphic, and several of them are pathogenic. They are considered the smallest self-replicating bacteria and can pass through a 0.2 micron bacteriological filter that can filter out all bacteria except mycoplasma. However, mycoplasma do have unusually tough membranes that are more resistant to rupture than other bacteria, since this cellular membrane has contained all the host factors. The presence of sterols in the membrane contribute to their durability by helping increase the forces that hold the membrane together. Other bacterial species occasionally mutate or respond to extreme nutritional condition by forming cells lacking cell wall, termed L forms. L forms, which lack cell wall, exhibit a distinct morphology compared to the original bacterial strain from where they are derived. Typically, L-form cells appear as spheres or spheroids, for instance, under phase contrast microscopy or transmission electron microscopy. L-form can arise from both gram-positive and gram-negative bacteria. However, during a gram stain, L-form consistently display gram-negative coloration due to their absence of a cell wall. The cell wall plays a crucial role in bacterial cell division, which predominantly occurs through binary fission. This process generally requires the presence of a cell wall along with the components of bacterial cytoskeleton like FTSZ or FIDC. The unique ability of L-form bacteria and mycoplasmas to grow and divide without these structure is quite remarkable. This may represent an early form of cell division that was significant in the evolution of life. This division process appears to involve the extension of thin projections from the cell's surface, which then pinch off to create new cells. The absence of cell wall in the L form leads to a less organized division, resulting in a diverse range of cell sizes from very small to quite large. Bacteria have two main types of cell wall structures. Gram-positive cell walls are relatively simple, featuring a thick layer of peptidoglycan along with teichoic acid, not shown in this slide, situated outside the plasma membrane. In contrast, gram-negative cell walls are more intricate, consisting of three layers: the inner membrane, a thin peptidoglycan layer, and the outer membrane that includes lipopolysaccharides. Cell walls contain a component known as peptidoglycan or murein. This unique substance has not been discovered anywhere else on Earth except in the cell walls of bacteria. Both types of bacterial cell walls also include other components, making them complex structures, especially when compared to the cell walls of eukaryotic microorganisms. The cell walls of eukaryotic microbes are generally made up of single substance such as cellulose found in algal cell walls or chitin in fungal cell walls. Peptidoglycan provides structural support to bacterial cells, forming a rigid layer that helps maintain cell shape and protect them against environmental stresses. The presence of peptidoglycan is a key factor in distinguishing bacterial cells from those of other organisms. Understanding the role of peptidoglycan not only aids in comprehending bacterial biology, but also has implications for medical research, particularly in developing antibiotics that target bacterial cell wall synthesis. By targeting this unique feature, scientists can devise strategies to combat bacterial infection, enhancing our ability to protect against disease caused by these pathogens. The bacterial cell wall serves multiple functions beyond providing structural strength to the cell. It plays a crucial role in maintaining the cell's shape, which is vital for its growth, reproduction, nutrient absorption, and movement. Additionally, it safeguards the cell against the osmotic lysis when transitioning between different environments or while taking in nutrients. Water can easily pass through both the cell membrane and the cell wall, which makes the cell susceptible to osmotic imbalances that could exert pressure on the relatively fragile plasma membrane. The cell wall also acts as a barrier to certain molecules like toxins, particularly in gram-negative bacteria. In the following slides, we will explore the molecular structure of the cell wall and examine how its composition influences gram-staining characteristic. Understanding the components of the cell wall is essential for determining whether bacteria are classified as gram-positive or gram-negative. This classification is crucial in microbiology, as it affects bacterial behavior, treatment options, and overall understanding of bacterial species. The primary element of the bacterial cell wall is known as peptidoglycan or murein, which is unique to the bacteria. In terms of structure, the peptidoglycan appears as a mesh-like layer. Each layer consists of long chains made up of alternating molecules of N-acetylglucosamine or NAG and N-acetylmuramic acid or NAM. These long chains provide significant two-dimensional tensile strength due to formation of peptide bridges like NAG and NAM within each layer of peptidoglycan. In gram-negative bacteria, tetrapeptide chains that originate from each NAM unit are directly cross-linked, while in gram-positive bacteria, these chains are connected by pentaglycine cross-bridges. Peptidoglycan subunits are synthesized inside the bacterial cell, then exported and assembled in layers, which contributes to the overall shape of the cell. Peptidoglycan is a complex carbohydrate made up of two derivatives of glucose, N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, which alternate in long chains. These chains are interconnected by a tetrapeptide that projects from NAM sugar molecule, creating a lattice-like framework. The tetrapeptide consists of 4 amino acids: L-alanine, L-glutamine, L-lysine, or mesodiaminopimelic acid, DPA, and D-alanine. The tetrapeptide can cross-link directly with the D-alanine of one tetrapeptide connected to the L-lysine or DPA of another. In many gram-positive bacteria, a crossbridge composed of 5 amino acids such as glycine, the peptide interbridge, links one tetrapeptide to the another. These cross-linking enhances the overall strength of the structure, with greater strength achieved through complete cross-linking, where every tetrapeptide is connected in some manner to another tetrapeptide on a different NAG-NAM chain. All natural amino acids we obtained are in the L form. However, the bacteria can produce the D form of the amino acid and use them to cross-link the cell wall peptidoglycan. D amino acids provides the bacteria with a significant advantage by offering protection against proteases, which are enzymes that digest proteins. Most proteases can recognize and break down the L form of the amino acid, but struggle with the D form. As a result, vertebrate proteases are generally unable to disrupt the cross-linking in peptidoglycan, which is crucial for bacterial cell wall integrity. This characteristic is especially beneficial for pathogenic bacteria, allowing them to evade the host immune response and thrive in hostile environment. The gram staining protocol is utilized to distinguish between two main types of cell wall structures. Gram-positive cells possess a cell wall made up of numerous layers of peptidoglycan, which can reach a thickness of 30 to 100 nanometer. This peptidoglycan is often interspersed with teichoic acid, which are carbohydrate chains that extend throughout and beyond the peptidoglycan layer. Teichoic acids may be covalently attached to peptidoglycan, known as wall teichoic acid or WTA, or linked to the cell membrane through a lipid anchor, in which case they are termed as lipoteichoic acid. Teichoic acid is thought to serve several vital functions for the cell, including the establishment of a net negative charge, which is crucial for creating a proton motive force. It also adds to the overall strength of the cell wall, which is essential for preserving the cell's shape, especially in rod-shaped bacteria. Furthermore, research indicates that teichoic acid may be involved in cell division by interacting with the machinery that synthesizes peptidoglycan. Additionally, teichoic acid seems to provide protection against challenging environments such as high temperature, elevated salt levels, and beta-lactam antibiotics like penicillin. Pathogenic gram-positive bacteria such as Streptococcus use a teichoic acid to increase their ability to cause infections. Since peptidoglycan is relatively porous, most substances can pass through gram-positive cell wall with little difficulty. But some nutrients are too large, requiring the cell wall to rely to use exoenzymes. These extracellular enzymes are made within the cell cytoplasm and then secreted past the cell membrane through the cell wall, where they function outside of the cell to break down large macromolecules into smaller components. The cell wall plays an important role in bacterial reproduction. In this slide, Left panel of the left figure shows the absence of beta-lactam antibiotics. A bacterial cell divides normally. In contrast, the right panel of the left figure shows a bacteria attempting to grow and divide in the presence of beta-lactam antibiotics. The bacteria fail to do so and instead shed their cell wall, forming osmotically fragile spheroplasts. The spheroplast form of the bacterial cell is osmotically fragile, killing the bacteria in a short period of time. Now, what are the beta-lactam antibiotics? They are antibiotics such as penicillins like ampicillin, amoxicillin, methicillin, piperacillin. In addition, cephalosporin, carbapenems, and monobactams are also beta-lactam antibiotics. We will study these in more detail in a later chapter. Penicillin-binding proteins are a category of proteins known for their ability to bind penicillin. These proteins are commonly found in many bacteria, and their name originates from the method of their discovery. Almost all beta-lactam antibiotics interact with PBPs, which play a critical role in the synthesis of bacterial cell wall. Penicillin-binding proteins play a crucial role in synthesizing cross-linked peptidoglycan from lipid intermediates and facilitating the removal of D-alanine from peptidoglycan precursors. Studies on the purified enzymes have demonstrated their ability to catalyze several reactions, including D-alanine carboxypeptidase, peptidoglycan transpeptidase, and peptidoglycan endopeptidase. In all examined bacterial species, these enzymes have been observed to catalyze more than one of these reactions. PBPs attach to beta-lactam antibiotics due to their resemblance in chemical structure to the building blocks that create peptidoglycan, as shown in the right panel. When penicillin binds to the PBP, the beta-lactam amide bond breaks, leading to a covalent connection with the serine residue at the active site of the PBPs. This reaction is irreversible, resulting in the enzyme's inactivation. The cell walls of gram-negative bacteria are more complex than that of gram-positive bacteria, with more ingredients overall. They do contain peptidoglycan as well, although only a couple of layers, representing 5 to 10% of the total cell wall. The significantly thinner layer of peptidoglycan is typically no thicker than 4 nanometer compared to gram-positive cells. Their cell envelope structure is also more intricate. Within gram-negative cells, a gel-like matrix filled in the periplasmic space situated between the cell wall and the plasma membrane. Additionally, there is a second lipid bilayer known as the outer membrane, which lies outside the peptidoglycan layer. This outer membrane is linked to peptidoglycan by murine lipoprotein or Brown's lipoprotein. The outer membrane is composed of a lipid bilayer, very similar to the composition of cell membrane with polar heads, fatty acid tails, and integral proteins. It differs from the cell membrane by the presence of large molecule known as lipopolysaccharides, which are anchored into the outer membrane and project from the cell into the environment. LPS is made up of 3 different components: the O antigen or O polysaccharide, which represents the outermost part of the structure, the core polysaccharide, and number 3, lipid A, which anchors the LPS into the outer membrane. The LPS is known to serve many different functions for the cell, such as contributing to the net negative charge for the cell, helping to stabilize the outer membrane, and providing protection from certain chemical substances by physically blocking access to other parts of the cell wall. The outer membrane does present an obstacle for the cell. While there are certain molecules it would like to keep out, such as antibiotics and toxic chemicals, there are nutrients that it would like to let in, and the additional lipid layer presents a formidable barrier. Large molecules are broken down by enzymes in order to allow them to get past the LPS. Instead of exoenzymes like gram-positive bacteria, the gram-negative bacteria utilize periplasmic enzymes that are stored in the periplasm. Where is the periplasm, you ask? It is the space located between the outer surface of the cell membrane and the inner surface of the outer membrane, and it contains the gram-negative peptidoglycan that we just discussed. Once the periplasmic enzymes have broken down nutrients down to the smaller molecule that can get past the LPS, they still need to be transported across the outer membrane, specifically the lipid bilayer. Gram-negative cells utilize porins, which are transmembrane proteins composed of a trimer of 3 subunits, which form a pore across the membrane. Some porins are nonspecific and transport any molecule that fits, while some porins are specific and only transport substances that they recognize by use of a binding site. Once across the outer membrane, in the periplasm, molecules work their way through the porous peptidoglycan layers before being transported by integral proteins across the cell membrane. In addition to peptidoglycan and teichoic acid, bacteria of the family Mycobacteriaceae have an external layer of waxy mycolic acid in their cell wall. These long-chain fatty acids contribute to the hydrophobic nature of the cell wall, making it resistant to desiccation and harsh environmental conditions. This lipid-rich barrier also impedes the penetration of certain dyes and antibiotics, which is why acid-fast bacteria retain the primary stain during the acid-fast staining procedure, appearing red when observed under a microscope. This distinctive feature is crucial for the survival of these bacteria in hostile environment and is a key factor in the pathogenicity of organisms like Mycobacterium tuberculosis, the causative agent of tuberculosis. Lipopolysaccharides refer to the components found in the outermost membrane of the cell envelope of gram-negative bacteria such as E. coli and Salmonella. These molecules share a common structure and design. Lipopolysaccharides are large entities composed of 3 main parts: one outer core polysaccharide known as the O antigen, the inner core polysaccharide, and the lipid A, which is primarily responsible for the toxicity associated with LPS. These components are all covalently linked. In modern usage, the term endotoxin is often used interchangeably with LPS, although a few endotoxins, in the original sense of toxin, residing in the bacterial cell that are released upon cell disintegration, are not related to LPS, such as the delta endotoxin protein produced by Bacillus thuringiensis. Lipopolysaccharides can significantly affect human health, mainly through their interaction with the immune system. LPS is a powerful activator of the immune system and acts as a pyrogen, meaning it can induce fever. In severe situations, LPS may provoke a vigorous immune response that can result in various types of acute organ failure, potentially leading to septic shock. Antigens in the O side chain can be identified through serological or immunological tests to detect specific pathogenic strain such as E. coli O157H7, a dangerous bacteria that leads to bloody diarrhea and kidney failure. Additionally, at lower levels and over prolonged periods, there is evidence that LPS may contribute to harmful effects related to autoimmunity, obesity, depression, and cellular senescence. Like bacteria, the archaeal cell wall is a semi-rigid structure designed to provide protection to the cell from the environment and from the internal cellular pressure. While the cell wall of bacteria typically contains peptidoglycan, that particular chemical is lacking in archaea. Instead, archaea display a wide array of cell wall types adapted for the environment of the organism. Some archaea lack a cell wall altogether. While it is not universal, a large number of archaea have proteinaceous S layer and is considered to be the part of the cell wall itself, unlike in bacteria where the S layer is a structure in addition to the cell wall. For some archaea, the S layer is the only cell wall component, while in others it is joined by additional ingredients. The archaeal S layer can be made into either protein or glycoprotein, often anchored into the plasma membrane of the cell. The proteins form a two-dimensional crystalline array with a smooth outer surface. A few S layers are composed of 2 different S layer proteins. While archaea lack peptidoglycan, a few contains a substance with a similar chemical structure known as pseudomurine. Instead of NAM or N-acetylmuramic acid, it contains N-acetyl allosamurinic acid or NAT linked to NAG or N-acetylglucosamine with peptide interbridges to increase the strength. So, in the bacteria, NAG and NAM are connected, whereas the pseudomurine, a NAT with a T, is connected to a NAG where NAM is missing. So that is why it is called the pseudomurine. Methanochondroitin is a cell wall polymer found in some archaeal cells, based in composition to the connective tissue component of chondroitin found in vertebrates. Some archaea have protein sheath composed of a lattice structure to an S layer. These cells are often found in a filamentous chains. However, the protein sheath encloses the entire chain as opposed to individual cells. Now let's take a look at some of the bacterial surface structures. Most prokaryotic cell possesses cell walls. However, some may feature additional structures outside the cell wall, including glycocalyces and S layers. The glycocalyx acts as a sugar coat and has two main types, capsules and slime layer. A capsule is a well-structured layer that sits outside the cell wall, typically made of polysaccharide or proteins. In contrast, a slime layer is less organized and loosely attached to the cell wall, making it easier to wash away. Slime layers can consist of polysaccharides, glycoproteins, or glycolipids. Glycocalyces enable cells to stick to surfaces, facilitating the development of biofilms, which are layers of microbial colonies on surfaces. In the environment, many microbes thrive in diverse communities within biofilms, as these structures provide a degree of protection. Biofilms typically retain water like a sponge, which helps prevent drying out. They also shield cells from being eaten by predators and reduce the effectiveness of antibiotics and disinfectants. While these characteristics benefit microbes within a biofilm, they pose challenges in clinical situations where the objective is often to eradicate microbes. The capability to produce a capsule can enhance a microbe's pathogenicity or its ability to cause disease, since the capsule make it harder for phagocytic cells, such as white blood cells, to engulf and destroy the microorganism. For instance, Streptococcus pneumoniae is known for producing a capsule that significantly contributes to its pathogenicity. As discussed in the staining microscopic specimen, capsules are challenging to stain for microscopy. Negative staining methods are commonly employed. S layers are a type of cell envelope structure made up of combination of structural proteins and glycoproteins. In the bacteria, it's located outside the cell wall, while in some archaea, they act as a cell wall itself. The precise role of S layer remains unclear, and they are challenging to investigate. However, existing evidence indicates that they may have various functions in different prokaryotic cells. These functions may include helping the cell to resist osmotic pressure and in certain pathogens engaging with the host immune system. In the slide, figure A shows the S layer in archaea with glycoprotein lattices as exclusive wall component are composed of mushroom-like subunits with pillar-like hydrophobic transmembrane domains, or B, lipid-modified glycoprotein subunits. Individual S layer can be composed of glycoproteins possessing both types of membrane anchoring mechanisms. Few archaea possesses a rigid wall layer, example pseudomurine, or in methanogenic organism, as intermediate layer between the plasma membrane and the S layer, in C. In gram-positive bacteria, D, the S layer glycoproteins are bound to the rigid peptidoglycan containing a layer via secondary cell wall polymers. In gram-negative bacteria, E, the S layer is closely associated with the lipopolysaccharide of the outer membrane. This slide presents several images of the S layer obtained through freeze-edge scanning electron microscopy, electron diffraction, and atomic force microscopy. On the right, you can see the result molecular structure of the S layer protein SBSB from Geobacillus. After discussing various imaging techniques in Chapter 2, you can now recognize how these state-of-art technologies can unveil the intricate details of microorganisms like never before. Many bacterial cells possess protein structure that are integrated into their cell envelopes and extend outward, enabling them to interact with their surroundings. These structures can adhere to various surfaces, facilitate DNA transfer, or assist in movement. The filamentous structures including fimbriae, pili, and flagella. Fimbriae are thin filamentous appendages that extend from the cell, often in tens of hundreds. They are composed of a pilin protein and are used by the cell to attach to the surfaces. They can be particularly important for pathogenic bacteria, which use them to attach to host tissues. Pili are very similar to Fimbriae. Some textbooks use the term interchangeably in that they are thin filamentous appendages that extend from the cell and are also made up of pilin proteins. Pili can be used for attachment as well to both surfaces and host cells, such as Neisseria gonorrhoeae cells that use their pili to grab onto sperm cells for the passage into the next human host. So, why would some researchers bother differentiating between Fimbriae and pili? Pili are typically longer than Fimbriae, with only 1 to represent on each cell. But that hardly seems enough to set the two structures apart. It really boils down to the fact that few specific pili participate in functions beyond attachment. The conjugative pili participate in the process known as conjugation, which allows for the transfer of small pieces of DNA from the donor cell to the recipient cell. The type 4 pili play a role in an unusual type of motility known as twitching motility, whereas a pilus attaches to a solid surface and then contracts, pulling the bacteria forward in a jerky motion. A flagellum, derived from the Latin word whip, is a hair-like appendage found in certain plant and animal sperm cells, fungal spore or zoospore, and various microorganisms, serving primarily for motility. Organisms known as flagellates are characterized by their possession of one or more flagella. For instance, gram-negative bacteria Helicobacter pylori utilizes its flagella to navigate through the stomach, allowing it to reach the mucous lining where it can colonize in the epithelium, potentially leading to gastritis, ulcers, and an increased risk of stomach cancer. Additionally, in some swarming bacteria, flagella may act as sensory organelles responding to external moisture. The structural and functional aspects of flagella vary across the three domains of life: bacteria, archaea, and eukaryota. Prokaryotic bacterial archaeal flagella run in a rotary movement, while eukaryotic flagella run in a bending whip-like movement. The prokaryotic flagellum uses a rotary motor, and the eukaryotic flagellum uses a complex sliding filament system. Eukaryotic flagella are ATP-driven, while prokaryotic flagella can be ATP-driven in archaea or proton-driven in bacteria. The bacterial flagellum consists of three primary parts: the filament, which is a long, slender structure that protrudes from the surface of the cell. It is made up of a protein called flagellin and is hollow inside. The production of flagellin proteins occurs in the cell cytoplasm, and these proteins are then transported across the cell membrane and wall. Unlike hair, which grows from the root, the bacterial flagellar filament elongates from its tip as more flagellin units are added until it reaches the desired length. A protein cap helps guide the flagellin units into their correct positions. The hook, the component, is a carved connector that links the filament to the flagellar motor. And the third is the motor. This is the rotary mechanism that extends through both the cell membrane and the wall, with extra parts of the outer membrane found in gram-negative bacteria. The motor comprises two main elements: the basal body, which facilitates rotation, and the stator, which generates the torque required for the rotation. The basal body features a central shaft encircled by protein rings, 2 rings in gram-positive bacteria and 4 in gram-negative bacteria. The stator is made up of motte proteins that encase the rings embedded in the cell membrane. Here is a video that shows the incredible molecular detail of bacterial flagellar motor. Various types of motile bacteria display different flagella arrangement. A bacteria with a single flagellum, usually positioned at one end of the cell, which is polar, is referred to as having monotrichous flagellum. An example of this is Vibrio cholera, the gram-negative bacterium responsible for cholera. Bacteria with amphitrichous flagella have flagellum or groups of flagella at both ends. An example is Spirillum minor, which causes Spiriary Asian rat bite fever, or Sudoku. Lophotrichous flagella are found in cells that have tuft at one end. The gram-negative Bacillus Pseudomonas aeruginosa, known for causing various infections including swimmer's ear and burn wound infection, has Lophotrichous flagella. Peritrichous flagella cover the entire surface of a bacterial cell, such as an E. coli, which exhibits a peritrichous arrangement. Directional movement relies on the flagellar configuration. Bacteria respond to various environment signals such as light, phototaxis, magnetic field, magnetotaxis, via magnetosomes, and most commonly chemical gradients, chemotaxis. Purposeful movement toward a chemical attractant like food or away from a repellent such as a toxic substance is achieved by lengthening runs and shortening tumbles. During a run, the flagella rotate counterclockwise, enabling the bacteria cell to advance. In a peritrichous bacterium, the flagella are bundled together in a streamlined fashion, promoting efficient movement. Conversely, when tumbling, flagella extend outward and rotate clockwise, creating a looping motion that hinders forward movement but reorients the cell towards the attractant. When an attractant is present, runs and tumbles still occur. However, runs are longer while tumbles are shorter, facilitating movement towards areas with higher concentration of the attractant. In the absence of a chemical gradient, the lengths of runs and tumbles become more equal, resulting in more random movement. Now that we have explored the fundamentals of bacterial flagellar motor and the mechanics of bacterial swimming, let's merge these two topics to discuss chemotaxis, among other types of movement. In chemotaxis, a beneficial substance like a nutrient is known as an attractant, where a harmful substance such as a toxin is termed as a repellent. When neither an attractant nor a repellent is present, the bacteria engages in a random walk, characterized by alternating between tumbles and runs, ultimately leading to the lack of specific direction. However, when a gradient is present, the bacterium's movement becomes biased, enabling it to gradually move toward the attractant and away from the repellents. So how does a bacteria determine its direction? Bacteria utilize protein receptor embedded in their membrane called chemoreceptors, which bind to specific molecules. This binding often leads to methylation or phosphorylation of chemoreceptor, initiating a complex protein pathway that affects the rotation of the flagellar motor. Bacteria perform temporal sensing, comparing the current concentration of a substance to the concentration measured seconds or microseconds earlier. This method allows them to gather information about the concentration gradient's orientation, such as a bacterium approaches in higher concentration of an attractant, the duration of the runs, driven by counterclockwise flagellar rotation, increases, while the tumbles, driven by clockwise flagellar rotation, decreases. Although the bacteria may occasionally move in the wrong direction due to tumbling, it will not stay off course for long. This biased random walk enables the bacteria to efficiently navigate up or down the gradient of an attractant or a repellent. Here is a clinical focus. Barbara is a 19-year-old college student living in a dormitory. In January, she experienced sore throat, headache, mild fever, chills, and a non-productive cough. She began taking an over-the-counter cold medication, but it did not alleviate her symptom. Although some symptoms improved, her cough and fever persisted, leaving her feeling tired and weak. The potential respiratory diseases responsible for her symptoms could include influenza, bronchitis, pneumonia, or tuberculosis. After several days of fever, congestion, and cough and worsening aches, Barbara suspected she had the flu and visited her university health center. The physician assistant informed her that her symptoms could be indicative of various conditions. During the examination, the PA noted Barbara's elevated heart rate and discovered she had hypoxemia using a pulse oximeter. He also heard crackling sounds in her lungs and noticed slight shortness of breath. A sputum sample revealed greenish mucus, and a chest radiograph displayed a shadow in her left lung, all signs suggestive pneumonia. The PA diagnosed Barbara with pneumonia and prescribed amoxicillin, a penicillin derivative. However, more than a week later, despite completing the full course of medication, Barbara still felt weak but managed her daily activities. During her follow-up visit, the PA considered that many bacteria, fungi, and viruses could cause pneumonia. Since amoxicillin did not resolve her symptom, he speculated the causative agent might lack peptidoglycan, indicating a virus, a fungus, or a bacterium that does not contain peptidoglycan. Another possibility was that the pathogen had developed resistance to amoxicillin. To definitely identify the cause of Barber's pneumonia, further diagnostic test would be necessary. Given the ineffectiveness of amoxicillin, the PA prescribed azithromycin, which targets a bacterial ribosome instead of peptidoglycan. After taking azithromycin as directed, Barbara's symptoms resolved and she began to feel better. It was concluded that the likely causative agent of her pneumonia was Mycoplasma pneumoniae, a prokaryotic bacterium that lacks a cell wall, thereby making it resistant to amoxicillin, which is a penicillin derivative. Thank you.